can't stand science right now.
Why not? Science is my favorite subject.
I just don't understand what we're learning. I don't understand anything, translation or transcription in a cell.
That's easy. I'll show you. Let's go to the classroom.
Hi YouTube, and welcome back to another episode of Science is Cool. Today we'll be learning about transcription and translation, also known as protein synthesis. At the end of this video, there will be a stop motion explaining what we're going to be learning. Okay, my hand motions are a little out of control. Okay, and transcription is a written or printed representation of something. And this is what happens to your body when your DNA copies. So we're going to go into the nucleus, and once we are there, we're going to make a copy of one section of the DNA, and this is now called mRNA, and the messenger RNA then leaves the nucleus and enters into a ribosome, and this is known as translation. In the ribosome, this is where the magic happens, a tRNA enters into the same ribosome and drops off an amino acid, and we hook these up with like the opposite base pairs. So, and out comes a protein, and this is protein synthesis. So here's the stop motion. There, you can put these back.
I understand it now.
Oh, another thing you might want to know about is mutation in DNA. There are three kinds of mutations. The first being point mutation. This is where a mistake has occurred in the DNA and nucleotides go back and fix this mistake. The second kind is in the insertions. This is when there's a single strand that breaks in a single location and when one of the strands loops out. The third one is deletion. This is when there's a break in two places. The two repair strands come together without filling in the missing nucleotides.
Science is really cool. This should make my next test really easy. Thanks, Allie.
Okay, we're about to go over what the mutations look like.
Okay, so this is an example of point mutation, and just a reminder, the base pairs are A, T, and GC. And point mutation is when the base pair doesn't have its right match. So this is an example of AC when it should be AT, and the nucleotide goes back to fix it.
Okay, so for this mutation, this is insertion, and this is where there's one break in the DNA strand, and it goes back and is fixed like a loop, like so, and the base pairs are still there.
This is deletion, and it breaks in two parts. Then it comes down and makes a bubble at the bottom.
What about you next?
It's not going to be all perfect now.
I don't understand science.
That explains a lot.
How tall are you?
Hi, and welcome back to Science is Cool.
This is the end of our video, and it's just thanks for watching.